Today, an FDA approval in prostate cancer, a new drug application submitted in CLL and Follicular Lymphoma, a clinical hold placed on trials of BPX-501, and promising findings in studies of melanoma, non-small cell lung cancer, and multiple myeloma. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved abiraterone acetate in combination with prednisone for patients with metastatic, high-risk, castration-sensitive prostate cancer. The approval was based on data from the Phase 3 Latitude trial, in which the addition of abiraterone and prednisone to androgen deprivation therapy compared with ADT alone led to a 38% reduction in the risk of death. After a median follow-up of 30.4 months, median overall survival was not yet reached with abiraterone versus 34.7 months with placebo for patients with high-risk metastatic castration-sensitive disease. The median radiographic progression-free survival with abiraterone was 33 months versus 14.8 months for ADT alone, representing a 53% reduction in the risk of progression or death. The OS rate at 3 years was 66% in the abiraterone group versus 49% with ADT. Abiraterone was first approved in 2011 in combination with prednisone for the treatment of men with metastatic castration-resistant prostate cancer following chemotherapy. Since then, it has gained approval earlier in the treatment paradigm for use prior to chemotherapy. A new drug application has been submitted to the FDA for duvelisib for full approval for the treatment of patients with relapsed refractory chronic lymphocytic leukemia and small lymphocytic lymphoma, as well as an accelerated approval for the treatment of patients with relapsed refractory follicular lymphoma. The application includes supporting data from both the Phase 3 DUO trial and the Phase 2 Dynamo study. In DUO, duvelisib reduced the risk of disease progression or death by 48% versus ofatumumab in patients with relapsed refractory CLL or SLL. In the overall study population, the median progression-free survival with the PI3K delta and gamma inhibitor was 3.4 months longer with duvelisib versus ofatumumab. Also, in the overall population, the overall response rate with duvelisib was 73.8% versus 45.3% with ofatumumab, and lymph node burden was reduced by more than half in 85% versus 16% of patients, respectively. In the intent-to-treat population, overall survival between the two arms was similar. In Dynamo, duvelisib demonstrated an overall response rate of 46% for patients with indolent non-Hodgkin lymphoma, including 41% in patients with follicular lymphoma. Also in Dynamo, the ORRs in the follicular lymphoma, SLL, and marginal zone lymphoma subgroups were 41%, 68%, and 33% respectively. At a median follow-up of 11.5 months, the median overall survival in the entire INHL population was 18.4 months, and the median PFS was 8.4 months. The FDA has halted 4 U.S. studies of BPX-501 for cancers and orphan-inherited blood disorders after 3 patients developed encephalopathy possibly related to treatment with the novel cellular immunotherapy. BPX-501 is an adjunct T-cell therapy administered after allogeneic hematopoietic stem cell transplant, comprising genetically modified donor T cells incorporating Bellicum's CASPA-CIDE safety switch. It is designed to provide a safety net to eliminate alloreactive BPX-501 T cells in the event of uncontrollable graft versus host disease. Bellicum Pharmaceuticals, the company manufacturing BPX-501, is currently conducting 4 Phase 1/2 clinical trials of BPX-501 in the United States. BP001 and BP005 of BPX-501 in adult patients with hematologic cancers following initial allogeneic HSCT. BP003, which is in children with orphan-inherited blood disorders, in which BPX-501 is administered after initial allogeneic HSCT, and BP008, which includes adults and children with blood cancers to treat post-transplant relapse and evaluate the potential for a titrated dose of remeducid to resolve uncontrolled GVHD while preserving BPX-501 cells. In a press release Bellicum Pharmaceuticals stated that the clinical hold does not affect its European BP004 trial exploring BPX-501 following initial allogeneic HSCT in children with hematological cancers or orphan inherited blood disorders. The company added that it has treated more than 240 patients with BPX-501 in 3 allogeneic haploidentical stem cell transplantation studies, and that it is awaiting formal communications from the FDA to determine the requirements for resuming studies. BPX-501 was granted an orphan drug designation by the FDA in February 2016. In melanoma, updated findings from the Columbus trial demonstrated that combination therapy with encorafenib and binimetinib improved survival versus single-agent vemurafenib in patients with BRAF mutant advanced unresectable or metastatic disease. The phase 3 data showed that the combination reduced the risk of death by 39% versus vemurafenib monotherapy, and the median overall survival was 33.6 months versus 16.9 months, respectively. The FDA is currently reviewing a new drug application supporting use of the encorafenib-binimetinib combination in this setting. Under the Prescription Drug User Fee Act, the FDA is scheduled to make its decision by June 30, 2018. The Columbus trial included 2 parts. In Part 1, patients were randomized in a 1:1:1 ratio to receive encorafenib plus binimetinib, encorafenib alone, or twice-daily vemurafenib. In Part 2, patients were randomized 3:1 to encorafenib plus binimetinib or encorafenib alone. Across the full study, the median progression-free survival for patients treated with 300 milligrams of encorafenib plus 45 milligrams of binimetinib was 12.9 months compared with 9.2 months for patients receiving encorafenib alone. And those who received a 450 milligram dose of encorafenib with 45 milligrams of binimetinib, the median PFS was 14.9 versus 7.3 months with vemurafenib. In non-small cell lung cancer, the immunotherapy combination of nivolumab and ipilimumab improved progression-free survival versus chemotherapy in treatment-naive patients with high tumor mutation burden disease. The preliminary findings stem from the open-label Phase 3 CheckMate 227 trial. Bristol Myers Squibb, the manufacturer of both immunotherapies, did not report any further data. CheckMate 227 is an open-label trial with more than 2,500 patients with both non-squamous and squamous histologies, evaluating frontline nivolumab-based regimens versus platinum doublet chemotherapy and first-line advanced NSCLC that is divided into 3 segments. In previously reported findings from the Phase 1 CheckMate 012 trial, at a median follow-up of 22 months for nivolumab monotherapy and 16 months for the combination cohorts, the confirmed objective response rate with nivolumab monotherapy was 23%, and the median duration of response was not reached. The confirmed ORR in the frontline setting for the combined nivolumab plus ipilimumab cohorts was 43%. The addition of pomalidomide to the combination of bortezomib and low-dose dexamethasone was associated with a significant improvement in progression-free survival in patients with relapsed refractory myeloma with prior exposure to lenalidomide. Findings of the Phase 3 Optimism trial were reported by Celgene, the manufacturer of pomalidomide. The company did not report any additional data, but noted that there were no new safety signals and that expanded results would be presented at an upcoming medical meeting. The international open-label Phase 3 trial randomized patients with relapsed refractory disease to the triplet of pomalidomide, bortezomib, and low-dose dexamethasone, or bortezomib and low-dose dexamethasone alone. The FDA granted an accelerated approval to pomalidomide as a treatment for patients with multiple myeloma following 2 prior therapies, including lenalidomide and bortezomib, in February 2013. This initial approval was based on results from the open-label Phase 2 MM002 trial. In this analysis, the overall response rate was 7.4% with pomalidomide alone and 29.2% in patients treated with pomalidomide plus low-dose dexamethasone. This week, we sat down with Dr. Andrea Sursek of Memorial Sloan Kettering Cancer Center to discuss early onset colorectal cancer.
There's a rising interest in early onset colorectal cancer, and this has to do with the fact that in the United States, as well as worldwide in developing countries, we have noted an increase in the incidence in patients under 50. So this is a patient population in whom we don't have screening guidelines. In fact, we don't recommend screening until 50 or older, and yet they are developing colorectal cancer. And some of the questions that arise, of course, are what are the predisposing factors? What's interesting about this population is that they are not all familial. So we know that patients with Lynch syndrome, patients with ulcerative colitis have an increased incidence of colorectal cancer in this young age group. But these patients don't have any of those predispositions. They don't even necessarily have any family history, or certainly not a family history of cancer in relatives younger than 50. So something is going on. There is very likely an environmental factor that is contributing to this rise. And what's interesting as well, and that some of the data that were presented at this meeting and also from the prior publications of the SEER database, is that these tumors appear to be predominantly left-sided, predominantly rectal. And we really don't know why that is, if there is perhaps a different driver, whether this is obesity or whether this is microbiome driven. Those are some of the research questions that are still ongoing and some of the things that were discussed at the meeting today. The major take-home point at this time for community oncologists, but even more so for internists and gastroenterologists and primary care providers, is not to dismiss symptoms of rectal bleeding, bloating, abdominal pain. That is how the majority of these patients present. That was clearly presented at today's meeting as well at GI ASCO. And so to see if these are the symptoms, to follow them up, to recommend a colonoscopy. At this time, I think it's too soon to say that we should do screening 45 and older or 40 and older, simply because although the incidence is on the rise, it's still low relatively to the population that would need to be screened. For community oncologists, the data suggests that these patients present with more aggressive tumors. There have been some data that they don't do as well. They present with later stage. It's really unclear whether they present with later stage because their disease is more aggressive or because they actually dismiss their symptoms until they develop advanced disease and then are diagnosed at a later stage. We don't know very much about how they respond to chemotherapy. Some of the data at today's meeting indicated that they actually do better per stage as compared to average onset, possibly due to improvement or a better ability to tolerate chemotherapy, possibly because their disease is more responsive. I think it's really a question of the biology, and at least what we've seen so far, including the analysis of the oncotype DX in early stage patients, there really didn't seem to be a very significant difference between the younger patients and the average onset who are in their late 60s or 70s. So I wouldn't recommend a change in treatment at this point for community oncologists. I think younger patients, from the data presented at today's meeting too, are treated a bit more aggressively, are more likely to get chemotherapy, but that is probably a combination of the oncologists trying to do what they can to treat the disease, and then also from the patients wanting the most aggressive, being very motivated and wanting the most aggressive treatment.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.